How to extract DNA from anything living. A lab. The purpose of this lab is to separate the DNA from a living source. The materials we will be using are the DNA source, a graduated cylinder, test tube label, liquid detergent, alcohol, meat tenderizer or pepsin, and a stirring rod. The first thing is to read the entire accompanying lab handout and answer the questions before proceeding any further. The questions are: why did the plant or animal DNA source need to be blended and strained? What is the purpose of the detergent? What are cell membranes made out of? How does an enzyme work? What does it break the DNA source into? And what is the purpose of the alcohol in this experiment? Next, select one of the DNA sources available for your experiment and use the graduated cylinder to measure 10 milliliters into a test tube. Label the test tube with your group number and which DNA source you used. Add an equal amount of liquid detergent to the DNA source and swirl to mix, but try not to make bubbles. Let this stand for 8 to 10 minutes. Add 1/4 teaspoon of meat tenderizer or pepsin to the test tube and stir gently to not break up the DNA, and wait another 8 to 10 minutes. The last step is to tilt the test tube and slowly pour 10 milliliters of alcohol into it to form a layer on top. Let it stand overnight. Hopefully, the DNA will float into the layer of alcohol. DNA is a long, stringy molecule. The salt that was added in step 1 helps it stick together. So what you see are clumps of tangled DNA molecules. DNA normally stays dissolved in water, but when salty DNA comes in contact with alcohol, it becomes undissolved. This is called precipitation. The physical force of the DNA clumping together as it precipitates pulls more strands along with it as it rises into the alcohol. If you want to save the DNA, you can transfer it.
Like a startling sign that fate had finally found me, and your voice was all